this video we will discuss about how glycans and immunity are related and how these glycans, prebiotic glycans, can be used to enhance immunity of the infant. So the immunity is here is presented as one of the factors that is with related to the this mucous gut thickness. So when there are no glycan structures are present, the thickness of this mucosa is less. That we can see here that some of the epithelial cells or gut epithelial cells are also removed, and this is not a healthy conditions. Whereas the healthy epithelial, this gut healthy mucosa is considered as a condition in which this gut epithelial cells has a high thickness, and this thickness can be increased or contributed by the glycan structures that we can see here. Here the thickness of the epithelial gut is high, and because of this it can interact with a whole lot of bacteria and generate an immune response and maintain a stable ecological network. Whereas in this case the bacteria can directly penetrate within those uh, gut epithelial cells and cause immunosuppressive reactions. So how this mucosa epithelial gut thickness can be increased? It can be increased by increasing the prebiotic mucin-like glycans in the food of the infant. That will leads to this prebiotic glycans, and this prebiotic microbial glycans then finally degraded and spread across these gut epithelial cells. And this is the zoom in view of how this one of the epithelial cell looks like. It has this glycan structures, and that can prevent this bacteria interactions directly to the gut epithelial cells. Whereas this interaction of this bacteria or epitome of this bacteria interacts with the glycan structure, that will trigger the immune response, and that will maintain a healthy immune system within the infant. So in this and the panel on the top right side represents some of the glycan structures that are found naturally in the human beings, and these are the mucin sugar coat. So glycan structures are made up of uh, mainly made up of these glycans, and these are some of the complex hypothetical mucin type glycans, and these are not usually found within the patient, but they can be formed in vitro in the labs. And these are some of the commonly found uh, commonly found glycans within the infant or within the human beings, that can be seen here. These are some of the small glycan structures. Some of them are O glycans, such as this galactose binds to this GalNAc through this beta 1,3 bond, and these things are formed. This formation of these glycan structures are possible because of this CAG enzyme, and that will interact with this kind of bacteria and maintain a healthy immune response. And another thing that has been mentioned in this paper is the relationship between this glycan structure complexity and this microbial diversity. And it has been proposed that after certain complexity, this microbial diversity will stabilize, and beyond this there will be no beyond this there will be no further increase in the microbial diversity. So this the optimum conditions or the optimum glycan complexity that is required to take care of this microbial diversity is been within this region, where this glycan complexity is enough to take care of the maximum number of the microbial diversity that are that human beings are presently exposed to. So this can help us to determine the uh, how much complex glycan structures or complex prebiotic glycans we need in our food, so that we can take care of the uh, we can take care of the infection that can be done by these microbes or bacteria, these different diverse bacteria. So I hope you got an idea how these glycans and immunity are interrelated. If you need to understand more about this glycan and immunity and this each how these complexities can determine the immune response of the immunity of the infant or in the human beings, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.